Hello everyone, and welcome to this presentation for the Digital Congress of Nanobiotechnology and Bioengineering. My name is Pablo Benici. I'm from the Regenerative Medicine Group at Aalborg University in Denmark. And my presentation will be about development of 3D bioprinted smooth muscle constructs. And first of all, I would like to thank the organizers for inviting me to this online event. I will be really happy to share my results with you and discuss some of the advances in this area. So first of all, I would like to introduce you to the research group I'm working in Denmark. Afterwards, because the main focus of my presentation will be on tissue engineering of a smooth muscle. I would like to recall some of the basic features of the smooth muscle and the tissue of smooth muscle. Then I would like you to recall some of the cells and techniques that are used in this field of tissue engineering of smooth muscle. To talk afterwards about the application of tissue engineering in urethral repair as a special case, and then how we are selecting and assessing different bioinks for bioprinting of smooth muscle constructs. And at the end, I will conclude with some perspectives of the field of bioprinting in general based on the results that we are having currently with our work on the urethral repair. So as I said before, first of all, I would like you to know a little bit about where I work. It's the group of regenerative medicine in Aalborg University. Before it was called the Laboratory for Stem Cell Research. We are doing research in stem cells, biomaterials, and probiotics, essentially for the regeneration or replacement of lost function. That's our vision. We are a group of about 12 people with one professor and several associate professors and assistant professors and some junior researchers consisting of PhDs and master students. We basically have a different amount of methods that we use in our research. Essentially, we have protocols for isolation of expansion of stem cells. Over the years have optimized a lot of these different procedures. But more importantly, we have expertise and methods and how to precondition these stem cells towards the different applications and applications. And to characterize the cells, we have also a lot of different biomolecular and cellular techniques that are used for characterization of these preconditions we do to the cells. So our research spans from basic research towards a clinical application of these stem cells. So we have some areas, for example, adipose-derived stem cells, some of the areas where we work in which we already have some applications towards the clinical side, for example, in chronic ulcers and osteoarthritis. And we have some other applications within the group in the field of probiotics, muscle progenitors, the one that I mostly focus on, and also some work about limbal epithelial cells for the treatment of corneal blindness. So the basic research is always about understanding how these cells behave, how to control the differentiation of these stem cells. In the, let's say, translational aspects of our research, we focus on how to translate these basic findings towards the use in humans, for example, by developing clinically compliant cell expansion procedures. And in the clinical application, of course, we focus more on the therapies in which the cells can be applied alone or in combination with some biomaterials, and then participate in some clinical trials. So in fact, our group has focused in the latest years in the topic of biofabrication, because we want to move forward from the classical two-dimensional cell culture towards more realistic physiological models of 3 dimensions. And in this respect, we are trying to investigate models, for example, for simple tissues like a urinary tract or the skin. Well, like when I call simple, I mean it's not so many, we don't involve so many cells in these models. Some models, they are models in which we investigate, for example, tissue development or the use of different therapies for wound healing, for instance. And towards the long-term goal, maybe to obtain through these biofabrication techniques, what we call organs on demand. So it's somehow very futuristic. And now when we see the topic of biofabrication, many of the authors focus so much on producing organs for replacements, but we see this as a long-term objective, perhaps to develop personalized implants or in vitro models. But nowadays we focus more on the in vitro models. We have different techniques for doing this. Basically, some biofabrication techniques based on bioprinters of different models, in which we can deliver the cells together with the gels, these materials, in order to produce 3-dimensional culture systems. Now, I said that my focus will be on smooth muscle or tissue engineering of smooth muscle, and therefore I would like to recapitulate some of the basic features of the smooth muscle tissue for those who are not familiar with this kind of tissue. So this basic the cells which we see in this histological section are basically some mononucleated cells, elongated, that have the capacity to respond to stimuli, like for example mechanical or chemical stimuli, and they contract in response to that stimuli. And they are mainly present in very places in the human body, for example in the skin, to contract the hair in the skin. Mostly in all the tracts, like the digestive tract and the urinary tract, in the hollow organs and the blood vessels, and also in the eye to control the contraction of the iris. So these are very important muscles which differ somehow to the skeletal muscle, because we call it smooth muscle because they don't have striation. So that's the main difference with the appearance of this under the microscope. And the contraction mechanism is a little bit different than the skeletal muscle, but it's also produced by sliding of actin and myosin filaments that when they slide against each other, they produce the shortening and the movement of, for example, a given organ or a part of the body that needs to be, where there needs to be the movement. So one of the main characteristics which I would like to highlight in this presentation is the phenotypic plasticity of the smooth muscle cells, these cells composing the smooth muscle tissue. The cells, as I mentioned before, they have this contractile capacity due to the presence of these myofibrils consisting of actin and myosin. And the cells, upon, for example, diverse stimuli, can switch to a so-called proliferative smooth muscle phenotype. And this is happening mostly in cases of pathological conditions, but also in some physiological conditions. For example, when women get pregnant, so there are changes in the phenotype of the smooth muscle in the uterus. So the main point here is that the cells have this phenotypic plasticity. There is a continuum between this contractile phenotype, where there is we call the fully differentiated smooth muscle cell, towards another phenotype in where there is a synthesis of different proteins that facilitate migration and proliferation of the cells. So they undergo mitosis and proliferate. But this, as I said, is mostly associated with some pathological conditions. However, when we have these cells and take the cells outside the body and put them in culture, they go through this process of differentiation, and they sorry, of de-differentiation, and they convert themselves in some sort of synthetic phenotype where they proliferate very actively. So, and this is very important for the field of tissue engineering, because most of the cells that we use are in this state, and we want to push them towards this more differentiated state when trying to develop tissue engineering applications. And so, as is mentioned here, under the effect of some growth factors or hormones, for instance, the cells will switch from the contractile to the proliferative state, but then some other growth factors are important towards the switch into the contractile phenotype. And these are important when developing strategies in tissue engineering. So there are many conditions in which we will require replacement or repair of smooth muscle tissue. Some of them are, for example, associated with the repair of hollow organs, for example, the bladder or the bowel in case of cancer. It can also be the esophagus or the stomach, so all other parts of the digestive system. There are other cases in which, for example, there has been damage to sphincters, which can lead to fecal or urinary incontinence after trauma, for example, in women after birth, after they give birth. So the thing is, this smooth muscle needs don't have such a great capacity to regenerate. So we need to find strategies to obtain sources of grafts, of tissue grafts that can be applied in these places where we have the damage. And it's also found in, for example, surgical repair of large blood vessels and the reconstruction of the urinary tract. This will be mostly the focus on my talk today. If we talk about which cells are relevant to explore when thinking about tissue engineering applications, primary, we will think about the smooth muscle cells that are the first choice, because they are cells that are normally resident in these smooth muscle layers. However, there are many studies that shown that the adipose-derived stem cells are an alternative source, very valuable alternative source of cells for generation of smooth muscle. Here we have some microscopic images of adipose-derived stem cells in comparison with the smooth muscle cells in culture. So when we use these cells, we would like to assess how well they could adapt or will transform themselves into the desired phenotype of contractile phenotype, because as I mentioned before, as soon as we put them in culture, these cells will lose their contractile phenotype. So we need to assess different approaches on how to manipulate this phenotype. So we can check basically the morphology of the cells that will tell us something about if the cells are synthetic or the contractile phenotype, but this is not very quantitative. We can just see if the cells are rhomboidal or elongated, so that will tell us if they are synthetic or contractile. However, we need to use more specific assays, and here we are talking about assays where we have immunofluorescent detection, for example, Western blotting, or molecular techniques such as qPCR. And then we use different markers, for example, smooth muscle actin, Caldes-Mono or calponin, also myosin heavy chain as a late marker of differentiation. So the cells that express these myosin heavy chains are the most differentiated cells. And there are also some other assays which must ensure that the cells have obtained the desired phenotype, and these are functional assays, for example, a gel contraction assay. And I will show you some examples here. For example, in this image, we can see a comparison between two cell types. As I mentioned before, human smooth muscle cells can be isolated, for example, from bladder biopsies. They can be expanded and afterwards can be induced, for example, in this medium that contains only a small amount of fetal bovine serum and heparin. So the cells, for example, here in the green color, you see the smooth muscle actin expression. And for the human adipose-derived stem cells, this requires a longer induction protocol. They require, first of all, induction towards the smooth myogenic phenotype, because these are stem cells that don't have any, I mean, they are multipotent, but they don't have any definition of their phenotype. So we have to first induce them for 2 weeks in medium containing this TGF-beta1 and bone morphogenetic protein 4. And after that period, we induce in the same medium containing heparin and low amounts of fetal bovine serum. And as you can see here, they also express these markers of the these smooth muscle actin markers. So, but there are other methods, as I mentioned before, like, for example, looking at the expression of these genes that are specific for smooth muscle, like smooth muscle actin, calponin, caldesmon, and myosin heavy chain. And in this study, for instance, we have investigated what is the effect of low oxygen tension on the differentiation of the cells. And the study showed that both the smooth muscle cell, primary smooth muscle cell from aorta, and human adipose-derived stem cells differentiate better and under 5% oxygen tension. This is under hypoxic conditions. The same study also observed the functionality of these cells, and also observed that under this 5% oxygen tension, the cells, both the smooth muscle cell from aorta and the human adipose-derived stem cells, produce a stronger contraction of these gels, showing that there is a larger or an enhanced functionality of the cells when they are differentiated under hypoxic conditions. So this could be, for instance, a strategy that could be used for helping the tissue engineering strategies, culturing and differentiating the cells in under low oxygen tension. We can see here yet another way we can assess the contractile properties in a response, for instance, to acetylcholine agonist carbachol. So in this series of images, maybe it's not that clear, but the idea is to measure how much the cells reduce in size. So here is the cell size over time in minutes, and you can see that the human aortic muscle stem cell, human aortic muscle cells reduce the size over time when stimulated with carbachol. This is the same for the adipose-derived stem cells that have been induced, but not for the stem cells that have not been induced. So it's clear here that the cells, upon stimulation and also preserving this low oxygen tension, become into the smooth muscle phenotype, being potentially of use for this tissue engineering of smooth muscle. Now I'm going to focus much more on a specific application in which we work, which is treatment of urethral stricture disease in men. As you might know or not, the urethra is just the conduit that transports the urine outside the body, both in men and women. But in men, there is especially problematic upon, for example, traumatic accidents or surgeries or injuries during, for example, medical intervention, that there is a damage into the lining of the urethra. And the problem is that there is a process of inflammation and further formation of scar tissue that reduces the lumen of the urethra. And that creates a series of complications. Sometimes these complications are so severe that it requires surgical intervention, and then it is required to use a graft material. The most common material used is a graft used from the buccal mucosa. So we restore the lumen of the urethra by using a piece of mucosa removed from the mouth. As well, it is important to focus not only in the mucosal surface, but it's also important to focus on the smooth muscle layer. As you can see here in this immunofluorescent image, this is a transversal section of the urethra in rabbit. The red color indicates smooth muscle actin. So there is the cells on the submucosal layer are rich in actin, in smooth muscle actin. They are mostly smooth muscle cells composing also the the corpus spongiosum here under this area of the urethra. So if you look at this, is a transversal section of the penis in the middle of that. So you have the corpus cavernosum, and then you have the corpus spongiosum around the urethra. So that's rich in smooth muscle cells, which provide support for the other cells and also are richly vascularized. So it's very important for mechanical properties and for support of the urethra, this smooth muscle layer. Therefore, we focus on that in reconstruction of urethra. So the current approaches for reconstruction, tissue engineering reconstruction, there are many investigations going on. They can be divided in two big areas. One of them, which is based on the use of scaffolds, so polymeric matrices of degradable materials. So without the use of any cells. And another approach, which is a little bit different, and it's mainly used when you need to replace longer strictures, which is called use of cell-laden constructs. And for that purpose, we can use, for example, 3D bioprinting. There are many factors that need to be taken in account when you produce a scaffold or an implant for urethral reconstruction. So far, there are different groups of signals. For example, the physical properties of these materials, also the structural properties, for example, how much pores the structure has, if there are several layers, or if there is only one layer, and also the nanoscale dimension. But most important of all is the biological performance. So all these materials need to be performed and be biocompatible and non-toxic, and ideally also be resorbable and with time disappear and be replaced by the cell's own extracellular matrix. So as for the first type of approach, we are currently using, for instance, scaffolds derived from biodegradable materials, like, for example, polylactic acid or chitosan. These materials are electrospun in different layers. This is a process in which we deposit fibers onto the surface, and these fibers are stacked in different layers, and combination of these materials could give rise to, for example, multi-layer scaffolds that you see here in this transversal scanning electron microscope image, where you have, for instance, in the middle a polylactic acid layer which gives the strength, and two layers of chitosan and polyethylene oxide that gives the cell-friendly properties for the scaffolds. So we are investigating this for urethral reconstruction, both in vivo and in vitro tests. One of the limitations of this strategy when you don't use cells is that then the cells have to penetrate the scaffolds once implanted. So this could somehow work well when you have short strictures where you have to replace the piece of the urethra, but when you have longer strictures and perhaps longer tissue defects, it's somehow more challenging for the cells to penetrate these scaffolds. So the idea with the other approach with the bioprinting is to obtain much larger concentration of cells at the same time that you fabricate the scaffold. So you can use materials in which you control the assembly and the maturation of the cells, especially these smooth muscle cells within the muscle layer. We have seen some examples in the literature where people has been investigating potential replacements based on bioprinted structures. Here you see it's some sort of tube done by printing, 3D printing polycaprolactone. This is a biocompatible polymer, biodegradable biocompatible polymer. And then using a fibrin gel, these different cell types, urothelial cells on one side and smooth muscle cells on the other side were printed. And these cells were investigated in vivo by the group of Zhang in 2017. Some of the things that this provide very interesting approach to fabricate a tubular structure and demonstrated that the cells survive over a long time. However, we are not sure if this could be applicable into as a graft material because it's somehow very rigid due to this core of the polycaprolactone. So we are currently using an approach that is purely based on the use of hydrogels without any hard material in the core. And for that purpose, we are having different hydrogels which are printed together with the smooth muscle cells, so that's they are bioprinted. We construct some 3D models of the urethra with this 3D printing. We mostly use UV-based cross-link, so our bioinks are cross-linked, so hardened by application of UV light. And then when we culture the cells for a certain amount of time, then we observe the phenotype by either microscopy or PCR assays, and we also assess the micromechanical properties. And then we are selecting what are the best performing materials and the best performing combinations to obtain a model of the urethra which can reproduce the maturation level that the cells have in vivo. Initially, we tried some of the most common gels using in bioprinting. This is alginate. It's derived from marine algae. This is a very versatile hydrogel. It's very easy to work with, and instead of using photo cross-linking, you use chemical cross-linking by immersing your construct in a calcium solution. It has a very low cost, and the cells have, as you can see here, the green represents viable cells, very high viability once you print the cells. The problem with this alginate, as many of you might know, is that this is based in a polysaccharide structure, so the cells do not have any attachment site for them to proliferate, and survive. So if these cells are attachment dependent over the time, they will not survive. So it's very low proliferation rate, especially for example for these smooth muscle cells. So we try to investigate some other options for printing the smooth muscle cells, and we enter into the use of collagen 1, which is a very a protein which is present almost in all tissues, and especially in the submucosal area of the urethra. This, of course, is much more suitable for adherent cells because it provides these sites of attachment for the fibronectin and for the integrins in the cells. And it promotes basically much more cell growth, as you can see in comparison with the alginate. This live ink, which is based in collagen, after only 3 days, there is much larger number of cells, and the cells also look much more elongated, meaning that the cells have attached and are spreading. One of the limitation of using the collagen-based bioinks is that they are quite expensive, and they also need a tight control of the temperature while bioprinting. So this might be a certain disadvantage, because if you don't control very tightly the temperature, so the shape, so the printability becomes a little bit poor. And one of the other limitations of this is that the mechanical properties are rather fixed over the time. However, we continue doing some studies in these collagen-based bioinks, and we observe how cells distributed over the time on these constructs, and they were mostly retained at the edges of our constructs, as you can see here. And then the cells also deposit some, this is an elastin staining, so you can see that there is some fibers of elastin were synthesized and distributed in the matrix of this construct. So all in all, it's a very positive result. Also, the expression of the genes controlling smooth muscle differentiation were quite high in all of these constructs, with exception of a smoothling. So it's so far it has been a very good result with the collagen 1 based bioinks, but as I said before, it's a bit expensive, and it takes a lot of optimization the parameters for bioprinting because it's of this high temperature dependence. We also investigated for the smooth muscle bioprinting the well-known gelatin methacrylate, which is based on, it's a derivative of collagen actually, is this gelatin. as we all might know. And this gelatin methacrylate has the advantage that can be cross-linked, so stiffened by using UV light. The promotion of cell proliferation and differentiation is also very good. It's very similar to what we obtain with the collagen. And what is nice is that we can tailor very fine the properties, the mechanical properties of these constructs by controlling the amount of time we expose the gels to the cross-linking light. Perhaps the most critical limitation of GELMA is that it's also very temperature dependent. You need to print a very specific temperature. Otherwise, if you move from that temperature a few degrees, so the printability becomes really poor. So one approach we are currently investigating is to mix this GELMA with alginate, because alginate has such a nice properties for bioprinting. So we combine photo cross-linking with chemical cross-linking. We have BioInk, which both promotes cell proliferation and differentiation, where the mechanical properties can be finely tuned, and it has much less dependence on temperature. So if you see this graph, these are rheological data from different blends of bioinks, so GELMA and alginate, and these steep curves correspond to pure alginate, meaning that the properties are changing a lot when we change the temperature. But as we increase the concentration of alginate into the mixture, so this curve becomes more flat, so meaning that the properties are quite stable over a wide range of temperatures. So we can obtain much better printability. And so you can see in this image, we have used different blends. These are 5 different type of blends over the course of 24 hours and over the course of, over immediately after printing and 14 days after printing. And we try to observe how this shape changes over the time. So by looking at this normalized cross-sectional area and comparing to a control, which is just a pure bioink, which doesn't have any value for bioprinting, but is like the perfect bioink like the perfect ink for printing. So we compare this the change in the shape over the time, and we find out, for example, that there is one group in one of these mixtures that attains the most stable and close to the control. This group number 4. And this group number 4 also has an elevated viability of the cells, something which we don't have in the groups where there is a high concentration of alginate. So over the time, we also observe that some of the blends have a better cell viability. And when talking about the number of cells, we counted the cells over the period of several days. And there are also some groups in which it's more favorable the proliferation in this combination of GELMA and alginate at certain ratios. And these ratios also show they are preferentially better for differentiation. So upon certain number of days where we have proliferated the cells, we induce them with this heparin and low fetal calf serum concentration. And then we observe that the cells, for example, in this case, attain a very high level of actin and a myosin heavy chain as compared to other groups, or for example, the group number 5, which is the pure pure GELMA. So all in all, in this study, we can conclude that the addition of alginate to GELMA stabilizes very much the mechanical properties against these changes in temperature, and especially improving the printability, so the shape becomes much more stable. We found that some of these blends are superior in terms of preserving the cell viability and proliferation, and that it's possible to find an optimal blend enhancing the smooth muscle myogenic differentiation. So this work is still in progress as part of a PhD study, and we are currently investigating the gene expression of these different types of cells and preparing a publication. So, but what will be next in perspective for development of bioinks? We have seen that there is a great variety of bioinks in the market. I only showed a few of them, but there are many more commercially available bioinks, and the problem is that they may have fixed properties, so they might have to adapt to the tissue of interest that you have. So in the future, maybe there will be need of novel inks, better than the existing commercially, where you can somehow be able to modulate cell behavior over time. This is a crucial thing. I believe it's going to be hot in the next few years. For example, could be bioinks that based on self-healing properties, so where the properties of these materials can, even if they change, for example, if the muscle contracts, it could be that these structures will break down due to this contraction of the muscle, but then the gel will have the property of regaining the initial property, so to allow, for instance, growth of the cells. So if the size of the construct needs to grow, so it will be without breaking down the main structure. So you can look at some more ideas about these self-healing hydrogels in this recent review. All in all, I have a take-home message for you that have been listening to my talk. And most of all, I want to give some general comments about the topic of bioprinting. We all know that it has a huge potential for many different tissue engineering applications. The main advantage of bioprinting is that it will allow the possibility of accurate delivery of cells together with the supporting material. So classical tissue engineer, where you have a scaffold and you put the cells afterwards, the cells need to penetrate the different layers, and that's somehow the challenge. With the bioprinting, you will be able to we are being able to print together the cells and the biomaterials. So perhaps in the beginning there has been so much hype about producing new organs from transplantation, but this is not the case. Nowadays we are focusing very much in development of in vitro models like the one I presented here, a model for the urethra, where we can investigate what are the different signals that control the assembly and the differentiation of the cells. And this is what I have presented, and the optimization process through finding the proper bioink in the process of developing this model. So the challenges right now are many. Perhaps some of them, which are the most important, is that we have a great variety of bioinks in the market. They are tested in some few applications, but each research group has specific needs. So optimization work is very resource demanding. So this is something we learn in the process. It's not like you just buy a bioink and uh, with these bioinks that are available, start working with your cells. You can be lucky. There are so many cells that are well proved with these bioinks, but there are some other cells that are less tested, and then you need to somehow optimize your own bioink. And the other thing I would like to stress is that the extrusion-based bioprinting, like the one we are using now, still has many limitations, because you need to control many variables at the same time to ensure a correct printability. So it's to preserve the shape of what you printed over the time, and to preserve also the density and the viability. So there are many bioinks that are very good to preserve these shapes, but on the other hand, they are not so good for the cells. But when they are compatible with the cells, somehow they are behaving a little bit poor with the cells. So that's why you need to find a balance, and you need to test your own bioinks in the process. That's the process that takes some time. So finally, I would like to acknowledge and say thanks to some of students, PhD students, who have been part of the work I presented here. And also the financial support we receive from different foundations to perform our work. And lastly, I would like to thank the organizers of the of this conference for inviting me to share my results. And feel free to contact me if you have any further questions about the lecture. Thank you.